Okay, so good afternoon, everybody, and welcome to this webinar about the MedPhab pilot line. The idea of this webinar is on one side to give you an update on the status of this European Union project, and also to give you some information about how the future of this project is going to be as we are reaching the end of the European project, and non-profit association is already planned and starting the first meetings. So you will receive information about the expectations for the future and how to contribute from external companies. The second goal is, of course, to promote the experiments that were running from companies who participate in the previous open calls. So we will have a nice set of speakers who will show some of the projects developed within the pilot line, and also the results and also the ideas that were developed within the European Union project. This is the agenda we have for today. So we will start with the description of the MedPhab pilot line and also the future association, as I mentioned. So we will discuss how to work with MedPhab and also how you can contribute from external companies in the future. And then we will proceed with 4 cases from the companies Surface Diagnostics, Aceptuba, SpectraCure, and Ensire. We will talk about cancer diagnosis, also cancer treatment, blood infections, and well, all the technologies that were implemented within these companies and the pilot line. So it's my pleasure to start the webinar today introducing you Cristina Lieder. She's the Executive Director at MedPhab Association, and as I mentioned, she's going to present the MedPhab Association. So Cristina, the floor is yours.
Thank you, Antonio, and good afternoon, everyone. Yeah, my name is Cristina Lieder, and I am Executive Director of the newly founded MedPhab Association. I'm connecting to this webinar from Finland, where we just got our first snow. I know many of you probably have still 20 degrees outside. In my presentation, I would like to introduce the MedPhab Association to you, and I will talk about the challenges in developing medical devices and how MedPhab can help you to face those challenges. And finally, I would like to tell you how you can collaborate with MedPhab. So the name MedPhab comes from medical photonics fabrication, and the MedPhab story begins year 2020 when EU granted funding to a project aiming to establish Europe's first pilot line dedicated to development, manufacturing, testing, and validation of photonic technologies in medical devices. The consortium partners of that project are presented here in the slide, and few of them are also giving a talk later today. But EU projects end usually after 3 and 4 years, and we wanted to keep the pilot line alive and sustainable, which is why 4 of the research organizations involved in the project, Tyndall from Ireland, VTT from Finland, Joneum from Austria, and CSEM from Switzerland, and 2 medical device manufacturing giants, Philips and Jabil, joined forces, and we formed a legal entity, an association called MedPhab. Photonics is gaining more popularity also in medical field as a key enabling technology to create improved diagnostics and therapeutic devices. And we will hear today great examples from my co-speakers today. And MedPhab's mission is to accelerate the uptake of those photonic innovations in various medical applications. Here is a list of common challenges that medical device developers can face. First, the medical devices are often very complex, and multidisciplinary know-how is needed to create integrated systems. And typically, this know-how cannot be found in one single company. Other major challenges comes from the business field itself, it takes considerable time and effort to be compliant with the medical device regulations. And thirdly, the photonic experts are scattered, and it might be very hard to find the right company or even a person that you need. And we have also seen, especially with startups, that there might be overestimation of the maturity level of the technology. And often the technology transfer to manufacturing has been started very late. And for these reasons, company may have underestimated how long it takes and how much it's going to cost to get their products to the market, and thus make unrealistic business plans. So how can MedPhab help with these challenges? First and foremost, MedPhab Partners employs hundreds of scientists, experts, and technicians ready to help with various technological challenges. They have state-of-the-art infrastructure to develop and manufacture photonic medical devices. And without going into details of this technology list that we can offer, I wish to highlight that our offering is very comprehensive, from photonic component development to integration of technologies to various platforms, including optics, electronics, and for example, microfluidics for point-of-care devices. We can also help with design and simulations, and of course, fabrication of prototypes, all the way to transfer them to a ISO 13485 compliant contract manufacturing. It is critical to consider manufacturing dependencies early. Often developers focus on putting their efforts in increasing the technology readiness level, but the product development team does not necessarily design the device manufacturing in mind. They focus on that the design outputs meet the design inputs, but maybe not consider how to manufacture those devices at scale with a competitive market price. And this is how we see as a high risk route. It's more recommendable to begin to think about the design transfer to manufacturing early, and this is where we can help. It is also worth remembering that FDA regulations of design control state that the product validation must be performed on initial product units, lots, and batches. And it can cause considerable delay and cost a lot of money if the contract manufacturer tells you that there's a problem with the producing your designed device, and redesign might be needed, and that again delays validation phase and market entry. So the collaboration with MedPhab always starts with evaluation of which phase the product development is and what is needed to get to the next phase, and hopefully all the way to these manufacturing phases 4 to 5. So basically having high manufacturing readiness level where contract manufacturing partner then takes over. As MedPhab's business strategy is to provide joint offering, we see MedPhab way of working as a critical part of how we operate. We have established operations for project management, design, engineering, equipment, supply chain, and production with phase-specific quality assurance. Documentation will be prepared with support customers' own quality management systems. Besides identification of project lifecycle phase and setting realistic requirements for the project, the customer will get realistic view on timelines, costs, technical feasibility, and potentially technology gaps, which help them in making realistic business plans. By this, customer will get accelerated design, development, and manufacturing services. MedPhab way of working also includes risk analysis for design and manufacturing process, which again reduces the risk to the customer. All MedPhab partners are trained in a way of working which allows seamless transfer of technologies from research organization to manufacturing partners and or to the customer. And hopefully some of you got interested and would like to work with us. And you can interact with MedPhab by contacting the front office that will direct you to the right partners. Any business agreements that will be done directly with these member organizations. And we have also commonly accepted legal documentation, so you don't have to make different agreements with different partners, which again saves the time and speeds up the initiation of the project. We have also gotten a lot of interest to join as a member of MedPhab. As a first step, anyone can join our LinkedIn profile and get updates on ongoing activities and where you can meet our experts. Second step is to become a collaborator. That means that you become a bit more closely connected with MedPhab. And depending on what type of collaboration you are, the company are taught the MedPhab way of working, and they can become part of the technology offering. And as a tip, our members are actually very successful in applying public funding. So a collaborator could be invited to join those proposals, which again, they can use then to finance their own R&D work. And becoming a full member is then a bit more demanding. Like I said, MedPhab is about offering joint development and execution of the project. So full members are generally technology providers. And we have approval process for full members to avoid any conflict of interest. And then to summarize, MedPhab aim is to provide accelerated market access for novel photonic medical devices with reduced development costs. MedPhab members have expertise and state-of-the-art infrastructure to develop and manufacture photonic medical devices, all the way from prototypes to certified manufacturing. And customers have access to this know-how through a single entry point, which is the MedPhab front office. And here you have the contact details through which you can get in contact with the front office. And I would like to thank you for your attention. And if you have any questions, don't hesitate to be in touch.
Thank you very much, Christina, for the interesting presentation about the way of working. We have the Q&A box where you can put all your questions. So do not hesitate to type in your question. I have one for you, Christina, and it's related to the future of the association. Perhaps you can give a few tips to the companies attending the meeting about the requirements to be a company that are member, let's say. What are you looking for? Because I understand that not all the technologies are interesting for the pilot line. Perhaps some of them are already covered by some of the members. Perhaps you can say two or three technologies that could be interesting for the future association.
Well, definitely, for example, partners that can provide, for example, clinical testing would be interesting and useful for our customers. Technology offering is very, very wide, obviously, as you saw from my slides, but we are in a process of identifying if there are any gaps that we might need. Definitely companies like Philips and Jabil are needed. The RTOs that we have on board already cover very big space. But yeah, so I encourage everyone to be in contact if you feel that you fit with the profile. Members, only thing is that we want to ensure that maybe end user companies are not very good fit for MedPhab, because we need to ensure that we can offer our services to everyone so that there is not these competing situations. So yeah, don't hesitate to be in contact, and we are happy to discuss how you fit in the MedPhab offering.
Good. Thank you very much for the recommendation. As I mentioned, we have this Q&A. We will have also more time for Q&A at the end of the meeting. I see already one question from Elizabeth Sibaker that is talking about the version of the webinar recorded. Yes, the answer is yes. It will be a on-demand option in the next days, so you can see the webinar later on, and well, you can see more in detail all the slides from Christina, for example, if you want to understand more in detail what is MedPhab about, and you will find also her contact details. So thank you again, Christina, for your time and your presentation. And I think we can move to the next speaker, that is the first experiment or the first success story that we have today from MedPhab Online. Is Val Noven. He is R&D project manager at Surface Diagnostics, and he's going to present the information about his photonic biosensor platform for cancer diagnosis. So Wout, whenever you're ready.
Yes, thank you very much. And thank you for the opportunity to present Surfix, the photonic biosensor platform, and the work that we did in the MedPhab project. So I will first introduce the company, and then I will go into the activities in MedPhab. So Surfix was founded as a spin-off from Wageningen University in the Netherlands, from the Department of Organic Chemistry. And for the first 8 years, Surfix focused on contract research and development, developing custom nanocoatings for their customers. And then in 2019, there was a big change in the shareholder situation, which was followed by an investment round. And then we started developing the photonic diagnostics platform that we are working on today, and that I will also be speaking about today. So since then, we have been developing our first product, and in parallel, we have been demonstrating the technology with some showcases, for example, on COVID-19, on IL-6, and you can, I think, in the notes with this webinar, you can find a paper about the COVID-19 work. And if you're interested, more white papers can be downloaded from the Surfix website. And earlier this year, we also started our first commercial development work, which deals with detection of bladder cancer from urine. So why do we want to focus on cancer? Well, if you look at the numbers, these ones provided by WHO, then they are quite staggering. Number of cases, number of deaths, costs associated, but also the potential for improvement is also huge. And we believe that a lot of this improvement can be realized by using liquid biopsy. So detecting biomarkers that are excreted from the cancer cells. These can be proteins or DNA. They go into the blood. You can detect them in the blood, or you can detect them in urine or in saliva or in some other bodily fluid. And this provides an easy, non-invasive, minimally invasive, and inexpensive way to get a lot of information from this tumor. And this can be useful all the way from screening programs until monitoring of treatments. But even before you can start addressing these applications, there's already a long road to be traveled, starting from discovery of the biomarker that you want to detect until you have a product that you can actually put into use in clinical practice. So the product that I will be talking about today is the first product that we are developing as Surfix Diagnostics, and it's aimed at assay development. So assuming that the biomarker is known, but then you want to develop an assay on this photonic biosensor platform that you can later further develop into a product for use in clinical practice. So in the pictures, you see the prototype of the assay development system that we're developing. You see there's a drawer where you can put liquids, you can put your reagents, your buffers, your sample liquids. There's a microfluidic cartridge that you put into the instrument, and of course there's a user interface that allows you to interact with the instrument. So this product is intended for assay development, and if you go to a next stage like assay validation or really clinical application, then you will probably need a different product that will look different, but it's based on the same photonic chip platform. So it means it also has the same benefits, meaning it has a very high sensitivity and specificity for detecting the biomarker that you want to detect. You can detect various biomarkers on a single chip, and these biomarkers can be both proteins or nucleic acids or DNA or RNA, and in principle you could even detect these on the same chip. And of course we want to make a product out of this that's easy to use for people working in clinical practice, so you don't have to have any knowledge of photonics or microfluidics to be able to run an assay and to get a result. So we're always trying to improve the sensitivity of our system, to expand the range of applications that we can cover. And as you see in the graph, over the past years we've already gained a lot in performance, both for immuno, so protein-based assays, and for molecular DNA-based assays. And we intend to keep on improving on this limit of detection. And if you want to do that, and you want to make products based on this technology, then you will find that it's going to be a highly interdisciplinary work that you're dealing with, and you have to combine a lot of different technologies. For example, you can look at the photonic chip itself. You can change its design to increase, let's say, the generic sensitivity of the platform. You can look at the biotechnology, so the assay development. So how are you going to capture and detect the biomarker of interest? You can do that maybe very sensitively, but it's also very important to prevent non-specific binding and non-specific signals that you can do by doing the right surface modification. So we develop nano coatings for that application. Also on the instrument side, there are a lot of things you can do in terms of reduce the acquisition and analysis of the data to reduce noise and drift, for example. And also on the cartridge side, there are many things to be investigated, and this cartridge is actually the topic of the rest of the presentation, because that's what we worked on in our MedPhab project. So the cartridge that we developed is a cartridge for our assay development system, and in the system, if you are developing an assay, you want to have maximum flexibility as a user to change the chip. So you want to be able to change the bioreceptors that you are immobilizing. You want to change the order of the bioreceptors. You want to be able to add positive and negative controls. So on the chip itself, you want to have a lot of freedom, but also in the way that you run your assay. So we call that an assay protocol or a recipe, meaning which liquids are going to be flowing over your chip, at which temperature, for which time, at which flow rate. So you want to provide the user with maximum flexibility, and that brings a number of design challenges when it comes to how you are going to design and make this cartridge. Well, for one, we are dealing with a photonic chip, so light needs to go in and out. So that's what you have to deal with. And the light source and detector are actually in the instrument, so you have to make sure that everything is well aligned, so the light gets into the chip from the light source and out of the chip to the detectors. If you're dealing with microfluidics, especially at elevated temperatures, you have to think of how you're going to deal with removal of air bubbles, so you have to do some degassing. And we want to offer maximum flexibility for the user, so he has to have access to the surface of the chip actually, and that makes it also interesting how you are going to seal the cartridge, because you can seal it, but then it's not accessible anymore in general. So this was the starting point of our MedPhab demo project. We were already working on this topic with CZM. And when we started the MedPhab project, we also got involved with Jabil. At the moment we started the project, we had already thought of a concept how we wanted to integrate the chip in the cartridge, and we already had a functional model. So some of the critical functionalities were there, but it was not yet a fully functional prototype. So actually the concept that we came up with was to make the cartridge out of two parts. One part we call the tray that has the photonic chip, and one part we call the lid that has all the fluidic structures, the degassing membrane, and the adhesive you need for sealing. But in the functional model that we had when we started the project, the degassing was actually not yet implemented, so that was something we had to design within the project. And when we started looking at the manufacturability, which was of course one of the main topics in the project, it turned out that we needed to make a change to the design of the tape, because the way we had thought of doing it in the first place turned out not to be really manufacturable. Also, when going from the functional model to a real prototype, we wanted to switch from micromilling to injection molding, and that also required some design changes to be made to the cartridge parts. And then, of course, with the help of Jabil, we spent a lot of time thinking about all the risks associated to the design and to the production process, the manufacturing process, and we had obviously to specify a lot of things, a lot of dimensions, a lot of tolerances, which were not all known at the beginning of the project. So that was it took us some time to get there, but in the end, we came up with the three partners with the production and assembly process that starts with injection molding of the cartridge parts. Then they are being coated, and this goes both for the cartridge parts themselves, but also for the photonic chip. And then the two parts of the cartridge, the tray and the lid, are being assembled. So the chip is being assembled into the tray, the degassing membrane and the adhesive tape are being assembled onto the lid. Then the two cartridge parts are ready to be packaged and to be shipped for use. So actually, when this first project was finished, we had the opportunity to continue the work in a second demo case project. And in this project that's still running, we are working to transfer the manufacturing process, the assembly process that was first implemented at CSEM to Surfix. So by the end of the second project, we should have a running assembly process at Surfix that will allow us to produce up to 20,000 assay development cartridges per year. And in this second project, we also added another partner to the consortium, that's VTT. So CCM is focusing on the technology transfer, and VTT is looking at how we can further automate the assembly of the lid part of the cartridge. And Jabil is looking at basically looking ahead if we are going to high volume manufacturing. Because as mentioned, cartridge that we are talking about now is assay development cartridge. So that's going to be relatively small numbers, but if you think about really clinical applications, then you're talking about much larger numbers. The cartridge will be more complex and it will have to provide the result to the user in a shorter time. And then in terms of numbers, we're talking about millions of cartridges per year. So we want to have some sort of a forecast on how this would affect the costs. And that therefore also, of course, the business case. So at the moment we're looking at some process improvements. So how can we do things more in parallel? How can we improve or increase the level of automation, and can we do more quality control in line? So these are all things that we are addressing in the second demo project. So looking at what we got out of this project, well, first of all, some very tangible results. So we received prototype cartridges, fully functional prototypes with the degassing membrane, with the chip inside, that we can use for verification and validation, not just of the cartridge itself, but also in combination with the instrument, which is of course very important. At the end of the second project, we hopefully have a running production process, and clearly we have increased the TRL and the MRL of this technology. And actually, I like that Christina already had the word reality check in her presentation, because that's also certainly what we got. So we now have a much better understanding of what we can expect in terms of costs versus volume, in terms of investments needed to set up production line, etc. But I think equally important as the tangible results are the things that we learned, and certainly as a startup, this project has helped us to create a lot more awareness and insight in what it actually means to go from design and testing and prototyping to really producing something. The level of detail, the number of parameters that have to be known, that have to be fixed, and all the things that you have to think about before you can actually start producing something and before you can actually start designing a production process. So in that sense, I can clearly conclude that the support from MedPhab has been very valuable for Surfix, and it also fits very nicely with the development phase that we are in. Christina also already mentioned some common pitfalls and beginner mistakes, and I think I can say that MedPhab has really helped us in going around some of these pitfalls. So it has been a very good experience for us. So with that, I would like to thank you for your attention. If there are any questions, I'm of course happy to answer.
Thank you very much, Wout. Again, you can type your questions in the Q&A box. I see already one question from Francesco Crisafi, but I think it's for Christina, so we can perhaps answer at the end of the meeting. And I can start with a question for you, Wout. You mentioned all the designs, changing the designs on the cartridge when going from the, let's say, initial idea to the prototype. Will these changes also affect the reader, affected the reader, or the cartridge itself can be modified and the reader is not affected, let's say, by the changes in the cartridge design?
Yeah. So of course we have some boundary conditions to like the dimensions of the cartridge, where certain interfaces are located, that we cannot change without having to change the instrument as well. these interfaces we kept intact. So within these boundary conditions, we can change things. So in this case, we did not have to adapt the reader to accommodate for the changes that we made to the cartridge.
And are you going to keep the design for the different biomarkers, or do you think for the future you will need to think again in different cartridge designs?
So currently we have on our photonic chip there are 6 sensor elements. So there are basically 6 sensors, independent sensors. So you can detect different analytes, or you can use them for positive, negative controls and things like that. So with this number of sensors, I think you can cover already a lot of applications. In principle, the sensor itself, the chip itself, can be changed. For example, to have more sensors on there. But certainly for the assay development part, I think with this cartridge, this chip and this reader, it's already very versatile, and it can be used for a lot of different applications. When you go to the next phase and you really are going to develop an IVD product out of it, then for sure the cartridge and the instrument will look different, because then the use case is completely different. You're not developing an assay anymore. You are running a fixed assay, and then, for example, you probably want to have your reagents on the cartridge and not in the instrument where you have to refill them. So then there will definitely be changes to both the cartridge and the instrument.
Okay, good. Thank you, Wout. So again, you can type your question in the box, and meanwhile we can move to the next speaker. That is Ishan Saikia. He is co-founder and CTO at Setuba, and he's going to present his project of the UV-based disinfection adapter. So Ishan, whenever you want.
Yeah. Am I audible? Yeah.
Good, Ishan.
Yeah. Can you hear me? Is it okay?
Everything
yeah.
okay.
And okay, and my screen is also live, I guess. Yeah. Okay. Hi, I'm Ishan, one of the co-founders of Setuba. We are a medical device startup based in Bern in Switzerland and Cambridge in the UK. And we have the vision to combat hospital-acquired infections. We have recently begun the MedPhab collaboration. As you can see on my screen, we have partners Tyndall in Ireland and Philips in Netherlands. So Tyndall is helping us to develop the optomechanical components, essentially figuring out the micro components, the mechanics, and the optical arrangement of the devices inside. And Philips, they're helping us with the process engineering and manufacturing of the adapters, such as injection molding and etc. So going back to what our fundamental problem that we are addressing is hospital-acquired infections. These infections, as you can see in my screen, comprises of a catheter, such as the central venous catheter. Such catheters are inserted into the patient in an ICU environment or for hematology oncology applications to administer drugs and fluids into the body. Such catheters stay in the body for a long time, for 10 to 14 days on average, could be more. And what happens, bacteria really accumulate in the entry wound, colonize the surface of the catheter, and due to slippage of the catheters, what might happen is bacteria get into the bloodstream and causing infections, such as known as CLUBC or CRUBC, which essentially stands for catheter-related bloodstream infections. Has a risk of 3 to 20% depending upon different conditions, such as the health and hygiene the skin biosystem of a patient, the immunocompromised or not. So different factors has a variability in the susceptibility of this infection. And what happens is that patient needs to stay longer just to get rid of this infection, maybe 10 to 14 days, during which there's additional nursing hours, drugs and medications, ICU operation costs, etc. And there have been different, strategies to mitigate such infections, such as sterile barrier, clean hand hygiene, microbial impregnated catheters. But definitely there has not been a, let's say, complete prevention. As you can see from the numbers, our target markets, such as in the Europe and the USA, they have about 3 million and 5 million central venous catheters used annually. And infections occur quite in the thousands, such as, as you can see in the numbers, and often leading to deaths in certain situations where sepsis develops and overpowers the patient. And unfortunately, in these cases, we have costs as well, over several billions, leading to a tremendous burden on the healthcare system. What we are going for is a strategy to use an alternate, approach, where we have a deep UV system around 220 nanometers. Why we use this system is because the wavelength has been found to be having a dual advantage of being germicidal, but yet non-harmful to healthy skin tissue. Typically, UV is known to have carcinogenic effect, especially 254 nanometers and over are, let's say, are advised to prevent direct, exposure to skin. However, when we go below, like 220 nanometers in the wavelength, it is mostly absorbed by the dead skin layer, as well as if it penetrates into the live skin tissue, the skin cells, the cytoplasm, or the surrounding material itself absorbs the UV, thereby not allowing the DNA mutation or DNA damage. So this has been well established by different studies as well. But what we're really going to develop in this MedPhab project is the adapter, as you can see on the screen. This adapter is a plug-and-play device, goes around the catheter, and is supported by an optical fiber that brings UV of this wavelength of a certain dosage in order to, homogeneously illuminate the area around the catheter. And it's automatic plug-and-play, and the nurses don't have to manually operate the device. Some of the basic, proof of concept studies we have finished over the past years with our collaboration in hospitals, as well as in microbiology institutes, where we have found, agreement with other studies regarding the germicidal efficacy. For example, here's a snapshot of Staphylococcus aureus as a bacteria, where a deep UV treatment that we administer has resulted in 3 to 4 log reduction of such bacteria in vitro. Similarly, for skin safety, we have already conducted studies for in vitro of lab-grown skin cultures, as well as biopsies from human patients, as well as animal samples, where we have illuminated to understand what is the if there is a carcinogenic effect or not. And what we have found that within the safe dosage that we're administering, which is also germicidal, we do not see any carcinogenic effect, thereby motivating us to move to the next step of really getting a product that is acceptable and that makes the users happy and really reduce the burden on the healthcare system. Some of these tests have been conducted in the previous years in Switzerland at the University Hospital Zurich. Moving on, we are actually now using our preliminary prototypes for usability studies as well in hospitals. We have our collaborator at the Inselspital in Bern, supported by Dr. Carmen Fortmüller, and we have a variety of these design changes and optimizations being introduced through our usability study. And this design optimization and the handling and human factor engineering directly feeds into the MedPhab project in order to develop the adapter that is going to be ready for let's say, animal studies in the next step. But as a precursor, what we have done so far is also through the support of Photon Hub in the previous studies, where we collaborated with the Vrije University in Brussels, where together with their help, we conducted simulations through ray tracing and figuring out the basic optomechanical components. And moving on from this stage where we have our prototype, we have to miniaturize it, get into a whole perspective of large scale OEM quantity manufacturability processes, the cost effectiveness, and what aspects need to be considered for really gaining a logistics and for transport and for really sterilization. So these are the aspects that we are going to address within the MedPhab project. And so what our overall roadmap is essentially to have contract R&D through our current, let's say, research programs with Philips and MedPhab and Tyndall, and then start contract manufacturing next year onwards, and hopefully make an entry towards FDA and UKCA eventually in these upcoming years of 2025 and 27. We are going through, potentially, we're exploring routes through the Breakthrough Device Program or the Safer Technologies Program in the FDA. And what really helps us to achieve these targets is essentially the MedPhab collaboration again, where our fundamental goals are, what we want to achieve is stage of technology readiness level 6, where we are able to demonstrate our performance in a relevant environment, which for us is in a step towards getting animal studies. Here we have exemplary swine model, where we have a catheter that is being currently in place for up to 14 days for administering different drugs and interventions. And what we want really out of this MedPhab project towards spring, that is when we aim, is to have a product functional prototype that can be placed around such a insertion site. And we are hoping or aiming for 3 such units initially of the functional prototypes, so that we are able to demonstrate safety as well as effectiveness in the animal model. This is in process towards finally going towards clinical trials in the hospital environment, in the clinical setting. And through this collaboration, we also have, let's say, eager customers who have been supportive of our journey so far. Some of our key opinion leaders comprise of leading hospitals in Switzerland, in Basel, and in Zurich. And further, we have collaborations, or let's say a market entry, let's say research happening together for cost effectiveness, value proposition, early economic health modeling in the NHS in the UK through NIHR supported funds. And in this case, we also like to welcome the broader member of today's audience to join us in this journey and follow our progress as we develop a disinfection device that is able to solve some of the critical problems that's persisting, leading to infections. And lastly, as I thank you for your attention, and I'm happy to take queries and questions and receive feedback in back. Yeah, so that's all from my side.
Thank you, Ishan. Again, I encourage you to type your question in the Q&A box. I have one question regarding the adapter that you presented. Is it working as a kind of light diffuser to cover the area you want to disinfect? Is there any other photonics phenomena involved?
Could you repeat the first part of your question? Is it, sorry, I did not get it.
Is a kind of light diffuser where you spread the light in the whole area of the skin?
Good question. So what we are going for is basically we are bringing light through optical fiber, and we are exploring different, let's say, concepts at the moment to how to uniformly illuminate the region. So some of the aspects that we are looking for is, optical fibers that have potentially tips that have a diffusing end, or could be optical fibers that are side emitting. They're thereby able to illuminate the region homogeneously. We're also exploring with different geometries of the let's say adapter itself, meaning a scattering surface or a reflective surface that is able to further homogenize the light that's been brought in by the optical fiber.
And are you also exploring filtering options to be sure that you are also only delivering the 220 nanometers wavelength?
That's a good question. So, considering the light sources, there are commercially available light sources that look towards, let's say, for example, for in vitro studies, we have an incoherent lamp that has a filter that basically cuts out any additional wavelength but lets 220 pass. But for our final product that's gonna have our light source, we are looking towards second harmonic generation light sources, which are basically focus on getting a light wavelength, for example, in the 400s down to 200s. The farther deep we go in the wavelength region, between 220 is our let's say more or less the window. We are not really precisely focusing on exactly 220 because deep UV has let's say the lower we go, we have the ability to have a higher dosage because the lower the wavelength, it gets better absorbed by the dead cell layers.
Okay, very good, very good. So also a challenge if any company wants to contact you, I guess you are interested in discussing options.
Yes,
so
like this MedPhab, like this MedPhab demo is focused on the outcoupling of light and really getting the adapter. But definitely for the incoupling of light or the light generation or light source development, we are looking towards on a separate, let's say independent, let's say product development aspect to have the most suitable light source that gives us the desired wavelength as well as the the power that we need to actually receive a germicidal effect. So the more optimal the source is, the better for us.
Very good, very good. So again, any question that you can have in mind in the next minutes, please type it in the Q&A, and we can have it at the end of the meeting. So thank you again, Ishan, for a nice presentation and also showing us some details of your development. And we can now move to our next speaker. We have Marcelo Soto Thompson. He's R&D specialist at SpectraCure, and he's going to present the idea for this prostate cancer treatment alternative. So Marcelo, the floor is yours.
Thank you very much. My name is Marcelo Soto Thompson, and I work as an R&D specialist at SpectraCure. It's a great opportunity for us to be able to present at this webinar, and I will be talking about how to transform a scientific idea into an actual cancer treatment, and we are focusing on prostate. SpectraCure is the name of the company, and we are developing prostate cancer treatment. We are located in Lund, Sweden. And if you can hear the trains, that is because I'm sitting right next to the train track. So I hope the traffic will be calm for the next 15 minutes or so. We are a total of 15 employees, and we're quite strongly focused on R&D. Most of us have a atomic physics background. So the idea was initiated at the Division of Atomic Physics at Lund University. Professor Stefan Andersson Engels was the group leader for the medical group team. Professor Katarina Svanberg was involved in a lot of the medical applications throughout the years, and her husband, Professor Sune Svanberg, who was the head of the department, has been driving many different ideas, and this was one of them. So photodynamic therapy research in Lund started already in 1982, so it's been quite a long journey at this point. And throughout this time, Professor Katarina Svanberg has treated approximately 30,000 cases of superficial tumors at the Department of Oncology in Lund. And of course, I'm extremely proud to be able to acknowledge Professor Anne Louis, who is actually last week was awarded the Nobel Prize in Physics, which is of course very exciting for us, as well as having a fifth female laureate. So our treatment is based on PDT, which is short for photodynamic therapy. It's a treatment modality in which you you inject the patient with a photosensitizer that makes the patient sensitive to light of a certain wavelength. We then use optical fibers that we insert into the tumor at various places based on ultrasound. We position the fibers at the correct position. And we start the treatment where we illuminate. In our case, we use 690 nanometers to activate the photosensitizer. And the photosensitizer then it will cause lesions around each fiber tip. We don't only use the fibers as a means of for treatment, but we also use them for diagnostic measurements, in which we use each fiber to illuminate, and the remaining fibers are used as detectors for the light. And using those measurements, we recompute the treatment time based on the tissue optical properties that we are receiving. So we recalculate the treatment time, and then we continue on until we have delivered the correct dose to the whole target prostate, as I said. So the benefits of photodynamic therapy are it's a local and very precise treatment, because you only activate the photosensitizer, the fiber tips basically, and you will have a treatment radius at up to approximately 1 cubic centimeter per fiber. And on the right side, you can actually see a picture from an actual treatment. The recovery is quite quick for PDT in comparison to other treatments. It spares connective tissue, which is important, for example, for the urethra, which is a sensitive area in the prostate as well. There is no open surgery as with many others, so there will be less complications due to that as well. The only thing is that we insert the needles, which are fairly big, but it's not a big thing compared to open surgery. And also the treatment is repeatable in comparison to radiation therapy, where you can actually receive your lifetime doses after a certain number of treatments. This is repeatable. And in theory, there is no upper limit to the number of times that you can be treated with PDT. I'm trying to move on with the slides. I don't know if something has happened. Okay. So this is a picture of our device as far. We have the hardware at the bottom, which it's a box that consists of the laser devices, and it's also accompanied with software for the dose planning platform. And if we take a look inside the device, this is our device. It contains what we call the photonics modules, which is actually our light This is our light engine, which consists of 3 lasers and 2 photodetectors, and they are coupled to a fiber coupled output. And at the moment, each of these take about an assembly time of about 8 hours, and unfortunately it contains a lot of manual steps with a lot of steps that actually require you to learn how to perform them in the correct way. And the part cost for each of these photonics modules is about 1,500 euros each. And keep in mind that each system contains 18 of these. So this is what we found was a very nice fit with the MedPhab project. In the MedPhab project, we are collaborating with Philips in the Netherlands. I saw some familiar faces in the other presentations, they help us with the mechanical design and the design for manufacturing of these photonics modules. And we also collaborate with CSAM in Switzerland, which are helping us, and they are helping us with the optical and the thermal simulations. And the project goals, as I stated before, is to reduce the assembly time and diminish or reduce the part cost, but also improve the manufacturability of this device. I've seen in the previous presentations lovely examples of how the manufacturability is actually something that I think our MedPhab collaborators are really good at. So this is what we have accomplished so far in this. We are in the second part of our MedPhab project, and our friends at Philips have developed a robotic assembly, and then the initial cost down of the mechanics was down by 24% in the first step. And for us as a company, we have the pleasure of working with highly skilled personnel that are extremely good in their respective fields and brings quite a lot of complementary knowledge to our company. And as also, I'd like to mention that we have gotten a much better understanding of the optical and also the thermal constraints of our device. I'd like, before I say thank you, I just want to highlight one timeline here, because I'd like to show you a little bit of the timeline for the company, and just to tell you as well a little bit of where we are at the moment. So the first publication of PDT was already back in 1987 here in Lund. It was earlier in some other places. And SpecProcure was founded 20 years ago when I was a PhD student at the division. And in 2010, the company was bought by private investors and was able to complete an IPO in 2015, which gave us the funding to start the clinical trial and the phase 1, which we are currently in. Well, there was a little bit of a hiccup during the clinical trial from 2019 to 2021, where pretty much all the clinical trials were stopped, during which time we were able to focus quite a lot on the technical part. And we were very fortunate to be able to be accepted as part of this MedPhab collaboration in last year. And it's continuing on at the moment. But I just wanted to show you a little bit of the timeline for the company. So it's been a long road. I haven't been on it for all the time, but for quite some time. And I'm really excited about this MedPhab collaboration. And having said that, I want to thank you all for giving me this opportunity to present a little bit of our company. Thank you very much.
Thank you very much, Marcelo, for the nice presentation of the project. I have a couple of questions, and one is related with the wavelengths that you are using for the treatment and for the diagnosis. I assume they are different. Can you tell us a little bit more about them?
Yes, like I said before, we are treating at 690 nanometers. That's basically a wavelength where you have a larger penetration in tissue. And then we do measurements of oxygen, because oxygen is required for the photodynamic therapy to happen. If there is no oxygen, which is supplied by the blood, there will be no photodynamic effect. So we also use wavelengths similar to pulse oxygenation to measure the oxygen content in the surrounding tissue. We also look at the fluorescence from the tissue, from the photosensitizer, to be able to determine the level of sensitizer in the surrounding tissue. Again, if there is no sensitizer, then there is no point in illuminating that section of the tissue. Or maybe you can wait for a little while for the sensitizer to replenish.
And regarding the fibers, did you explore different kind of fibers, different cores, numerical apertures? I don't know if you explore different options to have the best results.
We have looked into, well, the tissue is, as most of you perhaps know, is highly scattering. We're using flat fibers at the moment, but we are also looking into using diffusive fiber tips. Since we're using them as detectors as well, the diffusing tip cannot be, if the diffusing tip is too long, it's difficult to tell, to make the reconstruction of the oxygen and the fluorescence from. But we have looked into diffusing tips as well.
Okay, very good, very good. So as I mentioned before, any question that you can have in mind, please typing in the Q&A. And thank you again, Marcelo, for giving us this nice summary about your project and the future as well. And we can move now to our last speaker, that is Mark van Geffen. He's Manager Scientific Support at Inspire, and he's going to present us the journey from the idea to the prototype, the final product. So Mark, the floor is yours.
Thank you, Antonio, for the kind introduction. As said by Antonio, I'll take you through the journey from our idea to prototype to final product. And I work at Inspire, and the mission of Inspire is to bring the laboratory to the patient. On the right side of the DI, you see the Radboud UMC laboratory. It is equipped with a lot of systems, and the patient has to come to the hospital when he wants his blood diagnosed. But Inspire wants to offer another solution, shown on the left, the ENSI system. You see centrally, you see the ENSI pad, reusable reader that can insert the ENSI card inside. The ENSI card is the microfluidic cartridge that holds all the reagents and on which the blood is tested. The blood is obtained from the patient by the tap device. You can put the tap device on your forearm and punch the blue button. And then 30 microneedles will puncture your skin. And with the vacuum inside of the tap device, the vacuum will suck up the capillary blood and allows it to transfer it to the microfluidic cartridge. And ultimately, the mobile phone can read out the data and transfer it to the cloud. This system makes use of chemiluminescence. Chemiluminescent is making use of a compound aminoluciferin. It's coupled to, in our case, tripeptides that are specific, that can be cleaved by activated enzymes in the blood coagulation. upon activation, the aminoluciferin gets released, and by luciferase that is added to the microfluidic cartridge as well, and ATP and magnesium, the aminoluciferin is converted into light. And this allows that we can make this technology and use it as a platform. No light sources are needed, and therefore this allows us to miniaturize the system to a point of care device. And currently we have created a version A prototype for whole blood testing. This prototype has been verified and tested in the laboratory. From top to bottom, you see first a blood tube. A citrated blood tube is inserted in the blood connection, the blood connector, and that is connected to the black part. That's the housing or the envelope that houses the microfluidic cartridge. And this part is then inserted in the ENSI system. And this whole system is approximately the size of a lunchbox. And it differs a little bit. This product can be placed at the general practitioner and can be used by specialized nurses. So we want to also show you the housing in detail. So in the center, you see the housing opened up, and you look towards a syringe. The syringe pushes in some air in the blood tube, and that displaces the blood. And the blood is then via a needle placed on the white cylinder on the right top view. The white cylinder holds a filter. So the blood deposited on the filter, the red blood cells remain on the filter, and the blood plasma with the proteins pass through it inside the microfluidic cartridge. As a second step, the blister is pinched and activated, and buffer is released inside the microfluidic cartridge as well. And by capillary force, the buffer and the blood plasma mix and transfer through the cartridge and absorb as well spotted reagents like proteins and phospholipids that are deposited in the cartridge as well. And ultimately, the mixture ends up with 16 detection chambers with a volume of about 100 nanoliter. And these detection chambers are measured by spot sensors, 16 spot sensors, and these are placed in the sensor PCB. The sensor PCB is in the center of the slide in green and gold. That's the whole sensor PCB, and that contains the 16 spot sensors. And spot stands for single photon avalanche diode. And on the left bottom view, you can also see a gross representation of the microfluidic design, with again the spot sensor glued to the microfluidic cartridge. This is the version A setup that we have, and we want to produce and assemble the microfluidic cartridge in a pilot line. So in the next few slides, I will show the pilot line development and how it looks like. We obtained this pilot line via an EU REACT grant in collaboration with Demcon, Industrial Systems, Surfix, and Micronet. And in this grant, we employ the smart machine base of Demcon as a module to place various modules to assemble our microfluidic cartridge. So firstly, we want to coat our cartridge with an automated coater from the company Sonotech. On the left, you see the automated version, and on the right, you see an R&D type of coater we obtained from our neighbors that are in the same building as we are, and we obtained it more or less by serendipity. So now we also have a backup system for this automated coating system. The coating system will be delivered to Enzire next month, and then it will be installed. Additionally, we also have procured an automated spotting system from the company Synion. The proteins and the reagents and the substrates that we need for our assays and tests in the microfluidic cartridges are accurately deposited by the automated printer. Alongside, we also have a conveyor belt in this system that allows us to spot in total 240 cartridges in one run. Tomorrow, I will travel to Berlin to assess the factory acceptance test. And a few weeks later, the system will be installed in Nijmegen at our site, and the site acceptance test will take place. So these are the transport carriers that Synion designed and developed as well. On top, you see 12 microfluidic cartridges, and on the right, you see the layout of the cartridges. Please also note the two holes in this carrier. Of course, these holes are for the filter and the blister. Space is spared so the microfluidic cartridge can be placed in this holder. The carrier will also be equipped with an RFID, and this plate is also, you see the gray cylinders below the green base plate, and these gray cylinders allow for vacuum suction, so all the microfluidic cartridges will stay in place. And this carrier will be exchangeable to the coating system and the spotting system, the FlexiCoat and the S100. So we can transfer, and this allows for pilot production. So additionally, we also want to assemble the filter and the blister to the microfluidic cartridge, and that is done in this assembly kit that is seen on the left. In the central, you can see in the center of the station, you can see a product in feed, and on the right you see how the cartridge then follows the workstation. So first the cartridge will go to the left, and there's an ultrasonic welding station that welds either the filter on the left and the blister on the right, and then the cartridge will turn to the back side and to the right ultrasonic welding station as well. And then the second component is ultrasonically welded, and then it is arranged again to the front, and then an operator can take it out and collect it. So this again is a module that was procured in the EU REACT grant. So we also participate in the MedPhab program, and we are performing lamination trials in the MedPhab program. In collaboration with VTT in Oulu, Finland, we are evaluating whether our cartridges and the foil that we have chosen, whether that can also be automatically be laminated. So therefore, we will test the Delta converting line and the Datacon Evo pick and place line in Oulu. And based on the evaluation trials, VTT will propose a system and select the right machine builder to laminate our NZ cards. And after the project, we will then procure such a lamination system. And furthermore, we are also in the Netherlands participating in a growth fund application, or more specifically in the Next Gen High Tech program. And that also helps us and allows to upscale our in-house pilot production facility. We participate in a lab on chip project where our point of care NZ system is one of the 3 platforms that is worked on by approximately 60 other participants in this program. Surfix's platform is also one of the other platforms, and the CRISPR-Cas platform from the company Scope Diagnostics is the 3rd platform that is worked on. On the right, you see our spot sensor module. You see the 8 spot sensors, and in gray you see blinded spots. So that is to subtract the background. And in dark gray, in the center, you also see 2 temperature sensors to make sure that the blood plasma stays on a temperature of 37 degrees. That's mandatory since the enzymatic reactions in blood coagulation also take place at 37 degrees. So it's important that we control the temperature, and the spot sensor is helping us with that. So in the Next Gen High Tech program, we increase our competences in scientific arena, in regard to miniaturization, in design to manufacture. We will also start with clinical studies in the next quarter, and we have a strong focus on quality and regulatory. Of course, the Next Gen High Tech program is also focusing and allows us to identify our needs, and will allow us to work on our gaps. And we do this also with partners in the south of Nijmegen. We are located on the Noviotech campus with a lot of companies, familiar ones like maybe NXP and CITC. And we are also partnering with the pharmaceutical company Takeda already from the beginning and the start of Enzire in 2017. And other partners are located in Eindhoven or Enschede, only about 100 kilometers away from Enzire. And with these external partners, of course, we also have a great team comprising of development engineers, quality engineers, and since this year we also have set up an electronics engineering department, and we are currently with approximately 24 people. So we look forward to make the next steps in our point of care development. And if you have any further questions, I would like to give the floor back to Antonio so he can moderate the questions.
Thank you very much, Mark, also for presentation of all the stages of the project and all the fronts you opened during these years. I have a question regarding the pilot line you presented, and you are starting, but which challenges do you see for the near future? You need to work the repeatability, reproducibility of the cartridges. What do you think are going to be the next steps in terms of effort?
I showed you the start of the pilot line, but we still have to expand the pilot line and start to do our initial testing with that. So the next half year or year will have a strong focus on getting the pilot line working and get the first products out. And the products we get out of this pilot line will also need to function in clinical trials. So to really source that and to do some initial troubleshooting as well, I think that will be the main focus and the main challenge for the next year.
Very good, very good. Thank you for your answer. So as I mentioned, any question you can again type in the question and answers, and we can perhaps go to the final slides because MedPhab Pilot Line is going to be present in next CompaMed exhibition, CompaMed show during Medica. It's going to be 13-16 November 2023, and we will have a booth over there. So in case you are a company interested in discussing your challenges, your needs, for the development of a medical device based on photonic technologies, please pass by. We will have some demonstrators there, so physical examples of what we did, some use cases as well, and also the team of experts from MedPhab with Christina that they can discuss any challenge with you. And we can finish. I see one remaining question in the question and answers, and it was posted at the beginning of the meeting, and it's from Francesco Crisafi from Cambridge Raman Imaging. And I think it's for you, Christina, because he's asking what is the difference between the contract manufacturers and then the MedPhab, and why to choose one way or the other.
I wish I could go back to my slides. Maybe you remember there was a graph basically proposing 5 stages of development. Okay, we are almost here. Good. So this smaller image here on the right. So we have contract manufacturing companies as part of the MedPhab, but basically where we are focusing on is this kind of development road, which ends when the product is going into manufacturing. So we are offering services in this fast learning phase, quick trials, and then moving towards more and more higher level manufacturing readiness levels, which also brings these certain demands for medical device regulation, understanding, and documentation. So basically, we are somewhere in between academic research and contract manufacturing. That's where we have different operators. RTOs typically in this phase 1 and 2, and then 3 and 4 towards our contract manufacturing partners.
Good. So I think it answers the question from Francesco. And I don't see any other question, and we are close to the end. So I'm going to finish with this slide with the contacts of MedPhab. So we presented here the pilot line, the future association. Another question from Elena Letkaya from Philips. Manufacturing still can continue with the same partners as in MedPhab. The answer is yes. So again, we will finish with this slide with the contacts. So any comment, any question, any challenge that you want to discuss, we presented also, for example. So thank you very much to Marcelo, to Mark, to Wout, and to Ishan for showing their projects and challenges they are facing or they face it, the results they have obtained, the ideas. They are all of them very challenging, and I'm happy that they were awarded in the previous open call. So best of luck. And in these directions, please contact us in case you want to discuss any of your challenges or idea. And for my side, the thing is everything for today. This video will be on demand in the PMI website. So do not hesitate to be back and review all the presentations. And yeah, thank you everybody and good afternoon.